Welcome to This Explainer. I am so thrilled to have you here today because we're going to look at the fundamental unit of life in a completely new light. We usually think of the human cell as just some microscopic blob, but I want you to imagine it as a bustling, highly regulated, and incredibly complex metropolis. We're talking about a microscopic city complete with border walls, security checkpoints, power grids, and even waste management centers. By looking at it through this lens, we're going to untangle the massive, interconnected biochemical network that literally keeps you alive every single second of the day. So let's dive right into this. Here's our roadmap for today's explainer. We'll explore this cellular city by traveling through 5 distinct neighborhoods. First, the cellular border wall. Second, gates and security. Then we'll split our energy tour into Power Grid Part 1 and Power Grid Part 2. And finally, we'll clean things up at the recycling center. All right, let's start right at the city limits. Section 1, the cellular border wall, which in the cell is the lipid bilayer. Every city needs a secure perimeter, right? Now, this wall isn't just a static sheet of plastic. It's incredibly dynamic. Made of phospholipids, it actually features a distinct asymmetry between its inner and outer layers. When the cell needs a stiffer, more rigid wall, it uses saturated fatty acids, which pack together super tightly. But say the environment gets cold and the membrane needs to stay fluid, it'll incorporate unsaturated fatty acids with shorter tails to prevent all that tight packing. Plus, you've got cholesterol acting as this brilliant temperature buffer, just weaving itself between the lipids to increase or decrease membrane fluidity exactly as needed. Of course, any secure wall has checkpoints, and here, those are proteins. We have integral proteins that pass all the way through the membrane, and peripheral proteins that just sit on the surface. And this actually brilliantly illustrates why these proteins matter to us in everyday life. Let's talk about aspirin for a second. In the endoplasmic reticulum, there's a peripheral protein called prostaglandin H2 synthase. Its job is to take a molecule called arachidonic acid and convert it into prostaglandins, which are the very signals that cause inflammation, fever, and pain. Well, aspirin works by physically blocking the opening of this specific enzyme through a process called suicide inhibition. By totally shutting down that peripheral checkpoint, aspirin stops the pain signal right at its source. Moving on to our next neighborhood, Section 2, Gates and Security. We know the wall is secure, but how do essential supplies actually cross the border? And how does the city's alarm system function? These specific gates, the transporter proteins, are categorized entirely by how they move cargo. A uniporter is simple. It just allows a single type of molecule to passively diffuse down its concentration gradient. No energy needed. But when we need to do work, we use couple transport. A symporter couples the downhill flow of one molecule to drive a totally different molecule uphill in the same direction. An antiporter does the exact same energy coupling, but it moves the two molecules in opposite directions. So the key takeaway here is that the cell is literally using the energy of one molecule flowing naturally to force another molecule against its will. These gates also form the city's electrical alarm system, the nerve impulse, or action potential. Let's speed things up because this happens in milliseconds. First, the resting cell sits quietly at a negative 70 millivolts. Suddenly, a stimulus hits the threshold of negative 50 millivolts. Boom! Voltage-gated sodium channels snap open. Positive sodium ions rush into the cell, rapidly depolarizing the membrane all the way up to positive 40 millivolts. This electrical spike travels down the nerve like a wave. Immediately after, potassium channels open up, and positive potassium rushes out, plunging the cell way down to a hyperpolarized negative 100 millivolts, before the sodium-potassium pump slowly resets the whole system. Why should you care about this electrical rush? Well, if you've ever had a cavity filled, you have manipulated it. Local anesthetics work simply by passing through the cell membrane and physically blocking those voltage-gated sodium channels from the inside. If sodium can't rush into the cell, depolarization never happens. The action potential is completely halted in its tracks, meaning the nerve fails to transmit the pain signal to your brain. Now that we're inside the city, let's explore how it actually runs. Section 3, Power Grid Part 1. We're heading into the cytosol to explore the quick energy sector of cellular metabolism. Before we look at specific pathways, we really need to understand the 2 fundamental rules of the power grid. Catabolism is the breaking down of large molecules into smaller subunits, which releases energy. Think of it like burning coal. Anabolism is taking small molecules and building them up into larger complex structures. This requires an input of energy. And here's how this builds on our understanding of enzymes. Think of these next 2 enzymes as the on and off switches for the city. A kinase uses cellular energy, ATP, to add a phosphoryl group to a protein. A phosphatase does the exact opposite. It removes that phosphate group.
These reactions control almost every process we're about to discuss.
Okay, here is the very first step in energy production, glycolysis. Happening right in the cytosol, this is a 10-step catabolic journey where one 6-carbon glucose molecule gets broken down into two 3-carbon pyruvate molecules. Notice the energy accounting here. The cell actually has to invest 2 ATP up front, but it generates 4 ATP later on, leaving us with a net profit of 2 ATP. Now, the absolute most crucial step here is step 3, which is controlled by the enzyme phosphofructokinase, or PFK. PFK is the ultimate rate-limiting checkpoint. If the cell has high ATP, PFK slows down. If energy is low, PFK speeds up to burn more glucose.
Interestingly, glycolysis is also a target for defense. In step 9, an enzyme called enolase converts 2-phosphoglycerates to PEP. When you brush your teeth with toothpaste containing sodium fluoride, that fluoride competitively inhibits the enolase enzyme in the bacteria living in your mouth. By shutting down step 9, you literally starve the cavity-causing bacteria of the energy they need to produce tooth-destroying lactic acid.
So, glycolysis is great when you've just eaten a meal. But here's a question. What happens to the cellular city when it runs out of its primary fuel, glucose?
Well, the city relies on two clever backup systems, primarily managed by the liver. First up is glycogenolysis, which is the literal breakdown of stored glycogen into glucose 1-phosphate to feed back into the system. But if those glycogen reserves run dry, the liver shifts to gluconeogenesis to build brand new glucose molecules from scratch, using precursors like pyruvate and lactate. This requires basically reversing glycolysis, utilizing unique enzymes like PEPCK to bypass the strictly irreversible steps.
Section 4, Power Grid Part 2. Because, let's be honest, those 2 ATP from glycolysis, that's just pocket change. It's time to enter the city's nuclear power plant, the mitochondria. Those 2 pyruvate molecules we made in glycolysis are transported directly into the inner matrix of the mitochondria. There, they're converted into acetyl-CoA, which then feeds into this central loop, the citric acid cycle. Now, as this cycle spins, it does produce a tiny amount of energy, but its real purpose is to strip high-energy electrons off the carbon skeleton, capturing them in specialized carrier molecules called NADH and FADH2. Those carriers then bring their high-energy electrons to the electron transport chain, which is embedded in the inner mitochondrial membrane. The electrons are passed like hot potatoes across 4 protein complexes. As they move, their energy is used to pump protons out of the matrix, creating this massive pressurized gradient. Finally, those protons rush back in through complex 5, ATP synthase. This enzyme is quite literally a physical rotor. As protons flow through its channel, the head rotates, forcefully jamming ADP and phosphate together to manufacture massive amounts of ATP. Now, while we're deep inside this highly regulated city, it's vital to remember that it doesn't exist in a vacuum. It has neighbors. This biological harmony extends to your entire microbiome. Take Candida albicans, for example. It's a fungus that normally lives completely peacefully in your oral flora. But if the system's balance gets compromised, say by long-term antibiotics wiping out competing bacteria, or an immunocompromised state, this fungus can overgrow, leading to a nasty infection known as oral thrush. It's just a stark reminder that biological harmony is always a very delicate balance.
Returning to our power grid, let's put this into perspective with a staggering comparison. When oxygen is present, allowing the mitochondria's electron transport chain to run fully, the cell generates roughly 30 ATP from a single glucose molecule. Compare that to the mere 2 net ATP produced out in the cytosol by glycolysis. The mitochondria is absolutely the heart of the cellular city's power infrastructure.
Finally, we reach Section 5, the recycling center. Because when the city breaks down large structures like proteins, it has to deal with the waste. When proteins are broken down, we're left with amino acids. The cell splits these into a carbon skeleton, which can just be fed right into the power grid, and an amine group. The problem? That amine group quickly becomes ammonia, which is highly toxic. Here, the liver steps in as the ultimate recycling center. Through a process called the urea cycle, which spans across both mitochondria and the cytoplasm, hepatocytes safely convert that toxic ammonia into urea. The urea then safely travels through the blood to the kidneys, where it is expelled from the body. And this brings us to a final, rather provocative thought. We've seen how perfectly all these systems interlock. But what happens if just one pathway shuts down? For instance, what if carbon monoxide enters the system?
Carbon monoxide is deadly precisely because it binds to complex 4 of the electron transport chain, completely blocking electrons from ever reaching oxygen. The proton pumps stop, the ATP synthase rotor halts, and the city's power grid suffers a catastrophic blackout. It brilliantly illustrates how this microscopic metropolis is at once an absolute marvel of engineering and yet incredibly fragile. Thank you so much for joining me on this tour of the cell, and I hope this explainer leaves you marveling at the bustling biochemical city thriving inside you right now.